Hey everyone, Dr. Mungli here. So in this particular video, I will be explaining you a pyruvate carboxylase reaction and also a pyruvate carboxylase deficiency. Okay, let's begin. So what is pyruvate carboxylase enzyme? Now, the pyruvate carboxylase, as the name says, pyruvate carboxylase. Carboxylase is the last name of this particular enzyme. So as we know in the classification of enzymes, so enzymes that end as carboxylase, usually they belong to ligase class of enzymes. Okay. So what pyruvate carboxylase does? So the pyruvate carboxylase, it is going to convert pyruvate, which is a 3-carbon molecule, pyruvate, which is an end product of glycolysis. So pyruvate is converted into oxaloacetate, and oxaloacetate is a 4-carbon molecule. That means 3-carbon pyruvate is converted into 4-carbon oxaloacetate. That means an additional carbon will come from carbon dioxide. So carbon dioxide is incorporated into this reaction. That is why the name of the enzyme here is pyruvate carboxylase, because it is going to add carbon dioxide to pyruvate molecule. So in order to do this, so pyruvate carboxylase needs biotin. So biotin is a coenzyme for pyruvate carboxylase. You got to remember this. And also it needs ATP, because ATP is needed for as an energy source to incorporate carbon dioxide. And also it needs magnesium, Mg2 plus there. Okay. So in short, the requirement for pyruvate carboxylase, we can write it as A, B, C plus magnesium. That is A for ATP, B for biotin, C for carbon dioxide and magnesium there. Okay. So this is what is pyruvate carboxylase reaction is. And this pyruvate carboxylase enzyme, it is located in the matrix of mitochondria. That is where is the location of pyruvate carboxylase. So whenever pyruvate gets down into the matrix of mitochondria coming from the cytoplasm, so some of it is converted into oxaloacetate depending on the needs of the cell. Okay. So because you know oxaloacetate has to get into the TCA cycle to make citrate. So I'll write that here. Oxaloacetate, one of the fate of oxaloacetate is to go into citrate formation. Why? Because oxaloacetate in the TCA cycle, it will condense with the acetyl-CoA molecule to make a 6-carbon citrate. Okay, so that is TCA cycle there. So this is a part of TCA cycle. So citrate gets into the TCA cycle. That means pyruvate carboxylase enzyme is important for TCA cycle to get going. So that is you got to remember this. Now, oxaloacetate, one of the fate of oxaloacetate is oxaloacetate can undergo transamination reaction to make aspartate. So aspartate, amino acid aspartate can be synthesized from the transamination from oxaloacetate. So how the transamination occurs there? So it is alpha glutamate is converted into alpha ketoglutarate. So I write glutamate here. Glutamate is converted into alpha ketoglutarate, and that's when oxaloacetate is converted into aspartate, and this is a transamination reaction. Okay, so you got aspartate there. And also oxaloacetate is whenever person is in fasting condition, so say lactate converting into pyruvate or alanine converting into pyruvate, pyruvate has to go into glucose formation in gluconeogenesis process. So during that process, so pyruvate converted into oxaloacetate, oxaloacetate is converted into malate, malate is moving out of mitochondria into the cytoplasm, and in the cytoplasm malate is converted into oxaloacetate, and that's how it is going into phosphoenol pyruvate and all the way up to glucose formation. So that means pyruvate carboxylase enzyme is one of the important gluconeogenic enzymes. So that is why functioning of this enzyme is important for us, because it is going to take pyruvate into oxaloacetate, and oxaloacetate will go into glucose formation. So it can be converted into glucose. That's how pyruvate can be converted into glucose, and that is gluconeogenesis process. Gluconeogenesis process needs pyruvate carboxylase enzyme. Now let's move on to see what is pyruvate carboxylase deficiency. Now this pyruvate carboxylase enzyme, it is coded by a PC gene. So gene that codes for pyruvate carboxylase is a PC gene. What if this PC gene is mutated? So that will give rise to a mutated form of pyruvate carboxylase that decreases the activity of pyruvate carboxylase, and that is what is the name of the disease called pyruvate carboxylase deficiency. So pyruvate carboxylase deficiency, this is what is one of the disorders commonly asked in the exam in relation to gluconeogenesis process. Now let's see what happens if there is a deficiency of pyruvate carboxylase. Now if this enzyme, pyruvate carboxylase enzyme, decreases its function, so let's assume that this enzyme activity is decreased. So what will happen? What is the substrate for pyruvate carboxylase? It is the pyruvate. Product is oxaloacetate. That means there is an increase in pyruvate because this enzyme has decreased its activity. Substrate is going to increase pyruvate initially, and oxaloacetate will decrease. So there will be decrease in oxaloacetate. First, let's see what happens to this pyruvate which is accumulated, and then I'll come to what is the consequence of decreased oxaloacetate. Now, pyruvate that is accumulated in the mitochondria, because this is a mitochondrial enzyme here. So accumulated pyruvate will go back into the cytoplasm, and in the cytoplasm, this pyruvate, it will be converted into lactate. Pyruvate is converted into lactate by lactate dehydrogenase enzyme in the cytoplasm. That means when pyruvate is more, so that also leads to increase in lactate, and this will cause lactic acidosis. So a pyruvate carboxylase deficiency will lead to lactic acidosis. This is one of the signs that you are going to see in pyruvate carboxylase deficiency. And also note that what is the other fate of pyruvates other than going into lactate? Some of the pyruvates will undergo transamination reaction. So I have a video on transamination reaction. Link for that video is appearing in the right upper corner, and also I have given the link in the description below. If you are interested, you can look at transamination video and understand what exactly is transamination. So pyruvate undergo transamination to make alanine, and during this process glutamate is converted into alpha ketoglutarate. That's what is transamination done by alanine transaminase enzyme. Overall, what happens? Some of the pyruvate is directed towards this side, and that will lead to increase in alanine, and that is what causes hyperalaninemia. Hyperalaninemia means excess alanine levels in the blood is referred as hyperalaninemia. So far what we have seen, so pyruvate carboxylase deficiency, increase in pyruvate, thereby increase in lactate, that causes lactic acidosis. Increase in pyruvate causes increase in alanine, giving rise to hyperalaninemia. So make sure you remember this: excess lactic acid, excess alanine levels in the blood. You got to remember it could be because of pyruvate carboxylase deficiency. Now let's see what is the consequence of decreased oxaloacetate. Now, as I explained before, oxaloacetate is one of the molecule that is needed to take to continue citric acid cycle, because oxaloacetate combining with acetyl-CoA to make citrate, and citrate is part of TCA cycle. So what will happen to the TCA cycle? So when oxaloacetate is down, that means there is a decrease in TCA cycle, and the TCA cycle is decreased in a cell, say consider neurons or any other cell, there will be overall decrease in ATP levels. Okay, and that can lead to breakage of this muscle membrane or cell membrane. Any tissue can affect, because pyruvate carboxylase is present in the skeletal muscle, it is present in the neurons, so present in different tissues. So those tissues can, their ATP levels are decreased, so they can change the membrane permeability of the tissues. Like if you take a skeletal muscle being affected here, so skeletal myopathy can occur. So skeletal myopathy, and that can lead to breakage of the skeletal muscle and eventually giving rise to hyperammonemia. So hyperammonemia. Alright, so along with the hyperammonemia, so patients may have hypotonia. So these are some of the things that are because of decrease in oxaloacetate, giving rise to decrease in citrate, giving rise to decrease in ATP, giving rise to changes in the muscle membrane. Now, what is the effect of decrease in oxaloacetate in transamination reaction side here? So when oxaloacetate is down, so aspartate formation is also decreased, and aspartate, when it is decreased, overall it can lead to, later on it can lead to decrease in glutamate also. Now we need to consider that aspartate and glutamate, these are excitatory neurotransmitters. That means for the proper neuronal function, we really need to have sufficient aspartate and glutamate, especially NMDA receptors, they need glutamate here. So decreased availability of glutamate can lead to, eventually it can give rise to a mental retardation. That is one of the important signs that we are going to see in pyruvate carboxylase deficiency. Now going with oxaloacetate into glucose formation by gluconeogenesis process. You also remember now that under fasting condition, our blood glucose is maintained at normal concentration by gluconeogenesis process, and of course by glycogen degradation also. So the gluconeogenesis process, which needs conversion of pyruvate into oxaloacetate because lactate, alanines can be converted to glucose by making pyruvate. Pyruvate has to go into oxaloacetate, and then oxaloacetate will go into glucose formation. For that you need pyruvate carboxylase enzyme. So if this enzyme is deficient, oxaloacetate is decreased, that means gluconeogenesis process will decrease, and that will give rise to decrease in glucose, and that will cause fasting hypoglycemia. So fasting hypoglycemia is one of the signs that is seen in pyruvate carboxylase deficiency. Now let's put them all together. So pyruvate carboxylase deficiency, because of deficiency of PC gene, can give rise to lactic acidosis, give rise to hyperalaninemia, it will give rise to hyperammonemia, hypertonia, mental retardation, and fasting hypoglycemia. So remember all these signs, and if all these signs are mentioned in the case stem, it is a case of pyruvate carboxylase deficiency. Thanks for watching, and if you have any question related with this particular concept, so put that question down in the description, in the comment box. I will get back to you as soon as possible. Thank you.